Now, as we have discussed about the antigen and the antibody in our different lectures, it's time to discuss the antigen antibody reaction, which is a very important topic from exam point of view. So these antigen antibody reactions have been classified into two types based on their methods of demonstration. So earlier there were some conventional techniques to demonstrate the antigen antibody reactions, and those techniques were the precipitation reaction, the agglutination reaction, complement fixation test, and the neutralization test. So these were the four conventional techniques of antigen antibody reaction demonstration. Now with the changing medicine, with the modern medicine, there is evolution of some newer techniques to demonstrate the antigen antibody reaction. Those include the enzyme linked immunosorbent assay, the immunofluorescent assay, western blot, and there are some other evolving techniques also, newer techniques also, but we are not going into so much of depth. We have to just limit ourselves to these conventional reactions, conventional techniques, and ELISA, which is important from exam point of view and also from Viva point of view. So we will be reading these five topics, out of which here I will be discussing the precipitation reaction and the agglutination reaction. So first of all, talking about the precipitation reaction. So before starting the precipitation reaction, one should be very much clear about the antigen, that antigen can be a soluble antigen or a non-soluble antigen. If antigen is a soluble antigen, then when this soluble antigen reacts with its antibody at a particular temperature and pH, then there will be formation of insoluble antigen antibody complexes in the form of precipitates or flocculus. In such case, it is called as the precipitation reaction, because there is formation of precipitates or flocculus. While if the antigen is a non-soluble antigen, then when this non-soluble antigen reacts with its antibody at a particular temperature and pH, then there will be formation of agglutinates or clumps. In that case, that reaction will be called as the agglutination reaction. So that is the basic difference between the precipitation reaction and the agglutination reaction. In precipitation, the precipitation reaction is done by the soluble antigens, while the agglutination or the clumping reaction is done by the insoluble antigens. So from here we can derive the definition of the precipitation reaction as well. So see here, what I have written in the definition is that when a soluble antigen reacts with its antibody in the presence of electrolytes, optimal temperature and pH, then insoluble antigen antibody complexes are formed in the form of precipitates or flocculus, and this reaction is called as the precipitation reaction. This is a very easy definition. You can very easily understand and remember this definition. Okay, so why is this requirement of optimal temperature and pH? So this is because the solubility of the antigen may depend upon the temperature and pH. It may vary with the temperature and the pH. That's why we need a optimal temperature and pH for this reaction to be occurring. Now coming to the examples which may be asked in the vivas or even in the university exams. So first example we have for the precipitation reaction is the slide flocculation test. I hope you must have been remembering the syphilis class, where I had discussed about the VDRL test and the RPR test, that is the Viral Disease Research Laboratory test and the Rapid Plasma Reagent test, where one drop of antigen and the patient sera, which is supposedly containing the antibody against the syphilis, are mixed, and when they are mixed there is formation of flocculus, that is nothing but a precipitate formation. So that's why it comes under the it comes under the precipitation reaction, and hence it acts as an example for the precipitation reaction. Next example what we have for the precipitation reaction is our Ilex gel precipitation test that I have described in the diphtheria lecture. So what we do in the Ilex gel precipitation test is that we have a culture media. We take a strip which is impregnated with the diphtheria antitoxin, so that the antitoxin is spreading from this strip throughout the culture media. So these are all the antitoxin spreading out of the the impregnated filter paper. Now what we do, we make three streaks lines over that culture media which is perpendicular to the filter paper. The streak A is our taste strain streak, then streak B is our known toxigenic strain streak, and then the streak C is the known non-toxigenic strain streak. Okay, so what happens with all those streaks is that C, we know that the streak C is a non-toxigenic strain which we already know. Okay, so since it is non-toxigenic, that means it will not produce any toxin. That means there will be no toxin antitoxin reaction surrounding the streak line C. That means there will be no precipitate line seen around the streak line C. So see here, no precipitate lines are being seen in surrounding the streak C. So that is clear. And then we have streak B. Streak B is a toxigenic strain that is already known to us. So here we will see the streak line surrounding the streak line B. So see here, we are seeing the streak line surrounding the streak B, which are seen which are seen as the toxin and antitoxin reacts, and precipitation line is seen. So these are the precipitation lines surrounding the streak B. And then with the streak A, there may be streak precipitation lines or may not be precipitation line based on whether the taste strain is toxigenic strain or non-toxigenic strain. If it is a toxigenic strain, then there will be a streak there will be precipitation lines seen surrounding the streak A. If it is a non-toxigenic strain, then there will be no precipitation line surrounding the streak A, just like streak C. Okay, so thus these all lines that we are seeing surrounding the streak lines A, B, these are nothing but the precipitation lines. So this also acts as an example for the precipitation test. The next example that we have is the Ascoli's ring test, that is done to detect the anthrax antibody in the patient's serum. Now what do we do in this test is that we take a test tube. So we take a test tube and we put the patient's sera in it. So we take the patient's sera in it, which is supposedly containing the anthrax antibody, and then we put the antigen over the patient sera. We pour the antigen over the patient sera. So this is our antigen over the patient sera. Now what we see, if the patient serum is containing the antibody against the anthrax, then there will be formation of a ring at the junction or at the interface of the antigen and the patient's sera. This brown color ring is called as the Ascoli's ring. Okay, so this is nothing but a precipitate forming at the interface. So that's why it is also an example of the precipitation reaction. Okay, so this is all about the precipitation reaction. Now coming to the agglutination reaction. So I have described already that agglutination is shown by the insoluble antigens. So whenever a insoluble antigen is mixed with its antibody in presence of electrolytes, optimal temperature and pH, then clumping or agglutination is seen, and that is called as the agglutination reaction. Now these agglutination reaction are classified into two types, that is the direct agglutination or active agglutination test, and then the passive agglutination or indirect agglutination test. So before going into that, you should understand that antigen can be soluble or insoluble. Okay, as we have already discussed. Okay, so if it is insoluble, if it is insoluble, okay, if it is insoluble, then it will be directly showing the agglutination test. There is no problem in that, so that will be called as the direct agglutination test. And if antigen is soluble, then we can make this soluble antigen insoluble by adding some substances. Okay, so some substances, suppose substance S is added to this soluble antigen and we make it insoluble after that. So when this laboratory made insoluble antigen shows the agglutination test, then it is called as passive agglutination reaction, because here the soluble antigen is made insoluble with addition of some other substances from outside, and that insoluble antigen is then showing the agglutination reaction. So this type of agglutination will be called as the passive agglutination test. So I hope you now understand the difference between the direct agglutination test or direct or active agglutination test and the passive agglutination test. Now let me delete all these things so that it becomes clear now. Okay. Okay, so what is the active or direct agglutination test? So active or direct agglutination test is that agglutination test where antigen itself is particulate or insoluble. Okay, where the antigen itself is particulate or insoluble. Now, that direct agglutination test can be of two types. Either this can be done on slide or it can be done on tube. So, what is the importance of doing it in the slide or in the tube? So, main important importance or difference is that with the slide agglutination test, we can only identify certain things, but with the tube agglutination test, we can quantify the things. Okay, so in other words, we can say that the tube agglutination is a quantitative test. Okay, it's a quantitative, while the slide agglutination test is only a identitative test. We can only identify with the slide agglutination set. So how can we identify? What are the examples? So identification of the bacterial colonies is an example of slide coagulity slide agglutination test. So how is this so? So when we take a bacterial colony and put it on a slide, and then we make a particular antibody against a particular antigen of that bacteria, then there is clumping seen over the slide that identifies the bacterial colonies. So in this way, this helps in identification of the bacterial colony. Then we have blood grouping also, where we put one drop of blood of a patient on a glass slide, and then we add the anti-sera, and after that we see the clumping, then we can identify the blood group of that patient or that person as well. So here, yeah, while doing the blood grouping, we can identify the blood group. So that is also an example of the slide agglutination. Now coming to the tube agglutination. So tube agglutination is a quantitative test where we can identify, or we can find the quantity of the antibody or titer of the antibody in the patient sera. So in the tube agglutination test, what we do is we put a particular amount of antigen in different dilutions of the patient sera, so that we can get that titer of the antibody at which the agglutination reaction is seen. So in this way, we can quantify the antibody in the patient sera. Okay, so this tube agglutination is useful in the Vidal test, the Coombs test, Will-Flix reaction, and the Paul-Bunnell test. So these are all the examples of the tube agglutination tests. Please remember the examples. Now coming to the passive agglutination test. So passive agglutination, as I described earlier as well, that when a antigen is soluble, then we can make the antigen insoluble by adding some other substances like the latex, the RBCs, or the bentonite. These are the three substances which are discussed in different literatures. Okay, so we can add these any one of these three substances to make a antigen insoluble, and then that insoluble antigen can produce the agglutination reaction that will be called as the passive agglutination test. So one of which is the latex agglutination test. So latex agglutination test, where the latex is used to make the antigen insoluble. Some of the examples of such latex agglutination tests are the antibody, the anti-streptolysin O antibody detection, then CRP estimation, the RF factor estimation, and the beta HCG estimation. So in these examples, the latex agglutination test is used. These examples are very important from exam point of view. Okay. Now we will be discussing some of the special types of agglutination reactions, one of which is the heme agglutination test. If you uh, if you look into the name of this test, then you can derive the inference that it is something related to blood. Heme means blood. So heme agglutination test is a special type of agglutination reaction where we are utilizing the antigen of the RBC. We are utilizing the antigen on the RBC surface, okay, to identify some of the antibodies. Okay, So what are the different types of hemagglutination test? That is the direct hemagglutination test and the indirect hemagglutination test. So in the direct hemagglutination test, we are directly using the RBCs, okay, to detect some of the antibodies. So for example, we have the Paul Bunnell test. What is the Paul Bunnell test? So in the Paul Bunnell test, we are utilizing the SEEP RBCs to detect the Epstein-Barr virus antibody in the patient's sera. Okay, so with the SEEP RBCs, the Epstein-Barr virus antibody in a patient's sera can be identified. Similarly, in the cold agglutination test, the human RBCs are used to identify the mycoplasma antibody in the patient's sera. Similar is the Combs test and the blood grouping. So these are all the four examples of the direct heme agglutination test. While indirect heme agglutination is that that type of heme agglutination test in which antigen is made insoluble by aiding RBCs, so that the RBC attaches to that antigen and makes it insoluble or particulate, so that it can show the agglutination reaction. So in other words, we can say that it is a passive agglutination reaction. The indirect hemagglutination is a type of passive agglutination reaction. So I have written here also, the RBCs are used as carrier molecule to make the soluble antigen insoluble, and then it shows the agglutination reaction, that is called as the indirect heme agglutination reaction. Okay, so that's all about the heme agglutination test. Next we will be discussing the co-agglutination. So what happens in the co-agglutination, that we are utilizing here the protein A of the Staphylococcus aureus to detect antigens. Okay, you can understand a when you see the diagram here, that we have staph aureus here, and on the surface, on the cell wall of the staph aureus, we have protein A, which is bound to the FC portion of a IgG antibody, which has free FAB portions. Okay, so when this staph aureus with IgG antibody with its Fc portion being attached to protein A and Fab portion being free, that when reacts with the meningococcus bacteria or meningococcus antigen in the CSF specimen, then a clumping is seen, that is called as the co-agglutination. Co-agglutination means clumping occurring twice, okay, or the bounding occurring twice. So here the antibody is already bound once, and then again the antibody Fab portion binds with the the meningococcal antigen. That means two times binding occurs, and hence it is called as co-agglutination. Okay, so that's the meaning of the co-agglutination reaction. Now let's see the definition of the co-agglutination. So co-agglutination is when the protein A of Staphylococcus aureus, which is bound with the Fc portion of IgG with free Fab portion, is mixed with certain antigen. Here in our example, the antigen is the meningococcal antigen in the CSF. So when it binds to the certain antigen, then agglutination is seen. This is called as the agglutination reaction. Now in some books it is also referred to as the reverse passive agglutination test. Why is that so? So why it is being called as the reverse? So reverse because generally in the agglutination reaction we detect the antigens, sorry, we detect the antibodies. Okay, but here what we are seeing, that antigen is being detected using the antibodies bound to the protein A of the staph aureus. So here, since antigen is being detected in the CSF or in the serum or in the urine, so it is reverse of what is generally being done in the agglutination reactions. Okay, in all the agglutination reactions generally the antibody is detected, but here we are detecting the antigens, hence it is reverse. And why it is passive? Passive because the antibodies are not directly binding to the antigens in the serum or the CSF. Rather, the antibodies are first bound to the protein A of the staph aureus, then that makes them particulate, and then they bind to the antigens in different types of specimen, and then they cause the clumping and all. So that means it is passive, it is not direct, it is passive. So hence the name reverse passive agglutination test. Now, what is the use of this co-agglutination reaction? So the uses are that it may be used in diagnosis of different organisms in the body fluids. Like in serum, this co-agglutination reaction can be used to diagnose or detect the typhoid bacillus antigen. Then it in the CSF, it can be used to detect the meningococcal antigen, like in our this example. Then in the urine sample, it can be used to detect the gonococcus antigen. Now nowadays these tests are obsolete, because nowadays we have got some improved and better tests available for diagnosis of different antigens, I mean for detection of different antigens and diagnosis of different diseases. So we are no longer using these types of conventional and older methods. So that's all about the first part of the antigen antibody reaction. Okay.